Hello, my name is Deborah Chasman. I'm speaking to you today from UW-Madison. On behalf of my colleagues, I'm presenting our work entitled Breastfeeding and Farming Lifestyle Promote Predominant Bifidobacterium in Infants. Asthma is an immune-mediated chronic illness, and its prevalence is increasing worldwide. It is a lifelong disease, and treatment is primarily focused on symptom management. Development of asthma begins in very early life, but it isn't diagnosed until later in childhood. It is often preceded by conditions including allergic rhinitis, eczema, and wheezing. An emerging body of research has observed that people who grow up on farms have reduced rates of asthma and these immune-mediated diseases. As an example, these histograms show data from a study conducted here in Wisconsin. In red, children who grew up on farms have lower prevalence of these conditions compared to the non-farm in black. Several maternal and infant lifestyle practices have been associated with protection against disease development. They include close contact with farm animals and their stables, especially by milking cows, and frequent ingestion of unpasteurized cow's milk. The level of farm exposure also correlates with the degree of protection. People following traditional agrarian lifestyles experience lower rates of asthma compared to more industrialized farming communities. The Wisconsin Infant Study Cohort, or WISC, and Wisconsin Farm Study, WFS, are prospective birth cohort studies that aim to identify molecular contributors of farm exposures on development of asthma and childhood respiratory illness. Together, they consist of 3 study arms. The Wisconsin Farm Study arm of the project is comprised of infants from Wisconsin Amish families who follow a traditional agrarian lifestyle. I'll be referring to this group as TA. The Wisconsin Infant Study Cohort, WISC, includes infants from dairy farming families and non-farming families in central and upper Wisconsin. These studies recruited families who were expecting a child and followed the infants through the first 2 years of life, collecting health information and a broad range of environmental and personal biospecimens. Today, I'll be sharing an analysis of one of the data sets generated by the project. We are comparing the gut microbiomes at 2 months of age between the farm exposure groups. We performed whole genome shotgun metagenomic sequencing on 116 stool samples with participant characteristics shown in the table. All of the TA, traditional agrarian infants, were delivered vaginally and are exclusively breastfed, so we enriched for those categories when we selected non-TA samples. These whole genome sequence data will enable us not only to identify microbial species level information, but also functional capacities of the communities. We hypothesized that the microbial communities of the groups would vary with the level of farming exposure, and that the TA infants would harbor unique microbes compared to the non-TA infants. So let's start with a bird's eye view of the microbial community structure. We computed beta diversity from species level information using the Bray distance, and we also clustered the samples using Dirichlet multinomial mixtures to identify lane structure. The beta diversity plot on the left is annotated with the DMM cluster assignment, with cluster 1 in red, 2 in blue, and 3 in green. The plot in the middle is annotated with the farm group label for each infant, with TA in blue squares, farm in green triangles, non-farm in orange circles. The plot on the right is annotated by the infant's diet at the time that the sample was collected. Exclusively breastfed infants are blue stars, formula-fed infants in red circles, and those with the mixed diet of formula and breastfeeding are in yellow diamonds. These bar plots show the distribution of farm groups and infant diet within each DMM cluster. All but 2 of the TA infants fall into cluster 1, and all of the exclusively formula-fed infants fall into cluster 2. Clusters 1 and 3 are driven primarily by one Bifidobacterium species, longum and breve respectively, while cluster 2 has more diverse drivers with lower weights. In summary, the high-level structure of the data is driven by diet and also by farm group, specifically differentiating the traditional agrarian from the non-TA infants. Now let's take a look at the organisms. These stacked plots show the relative abundance of top genera aggregated by farm group and diet. The bars on the left show exclusively breastfeeding infants, in the middle, those with mixed diet, and on the right, the exclusively formula-fed. The dominant genus across most of these categories is Bifidobacterium, which is bright yellow. While all of the exclusively breastfed infants and the mixed diet infants have high Bifidobacterium, the TA infants have relatively higher Bifidobacterium and also relatively lower diversity of other organisms. We also see other patterns associated with diet. For example, infants with any formula have more Blautia, which is this light blue bar, and the very few exclusively formula-fed infants that were included in the study have a lower abundance of Bifidobacterium and a higher abundance of Streptococcus, which is dark red. We compared the samples based on species level alpha diversity. Here are two example metrics that are significantly associated with diet and with farm group independent of diet. We observed a pattern that the TA infants had lower diversity but higher dominance metrics, which summarize the relative contribution of the most abundant species. So next we wanted to know which Bifidobacterium species made up the genus level totals. This bar plot includes only exclusively breastfeeding infants with any detected Bifidobacterium and shows the average distribution of Bifidobacterium species. We observed that the TA infants are predominantly colonized by longum, which is shown in light blue, while the farm and non-farm have a greater diversity of species. So for this data set, the diversity metrics are capturing this predominance of Bifidobacterium longum in the TA samples. Our next question was to survey the genetic diversity at a more granular level. We performed a pangenome analysis using PanFLAN to survey which Bifidobacterium genes were present in each sample and to compare them to reference genomes. Each row in this heat map represents a UniRef90 gene family that is found in at least one publicly available reference genome for Bifidobacterium longum. Each column is either a study sample or a reference genome. Red indicates presence of a gene, and orange indicates absence of a gene in that sample. The first bottom annotation indicates the reference genomes in light blue and the study samples by their farm group with TA in dark blue, farm in orange and non-farm, farm in green and non-farm in orange. The second bottom annotation shows the study samples by diet with exclusively breastfeeding in dark blue. And at the bottom for reference genomes, we include the subspecies annotation if it was available. Subspecies infantis are shown in pink, suis are in green, and longum are in blue. Using hierarchical clustering to compare the gene family representation in our study samples to reference genomes, this heat map shows that the TA samples cluster next to known infantis strains. The purple box here is highlighting the TA study samples, and the pink box is highlighting the infantis strains. We also see some genetic differences between the TA study samples and the infantis strains, suggesting that the TA samples may have some different functional capacity compared to the available infantis reference genomes. Finding more similarity between infantis and the TA study samples compared to the non-TA samples is consistent with a body of work that has observed a decline in infantis prevalence in cities and Western lifestyles compared to traditional agrarian communities. Bifidobacterium infantis in particular is interesting because it has a full complement of genes for metabolizing human milk oligosaccharides and other components of breast milk, whereas other related species have subsets of those genes, although they can perform cross-feeding. We further confirmed that the study samples have greater prevalence of HMO metabolism genes compared to the non-TA. The second heat map shows a subset of HMO genes that are found in the reference files packaged with human 3. The top half of genes are found broadly in Bifidobacterium longum, different subspecies, and we also see that they're prevalent in both the TA and non-TA study samples. Well, the bottom half of the genes are prevalent in our TA samples, not so prevalent in our non-TA samples, and these genes are specific to infantis. We broadened our analysis by comparing the genetic capacities for metabolic pathways between the groups. This heat map summarizes the significant pathways where we're looking at the intersection of hits between two different statistical tests, Moslin and Limma. One of the most significant pathways was folate transformations 2, which pertains to B vitamin metabolism. The TA infants have a higher abundance of reads for this pathway compared to the non-TA infants. Both bacteria and humans need these B vitamins for development. We also see differences in pathways for short-chain fatty acids and amino acid metabolism, including aromatic amino acid metabolism. We were interested in what other organisms and interactions among organisms distinguish the groups. So we trained machine learning models and observed that it was possible to distinguish the TA samples with high accuracy. These box plots are showing machine learning method performance for 10 repeats of tenfold cross-validation, where the top row of plots shows the area under the precision recall curve, and the bottom row shows area under the receiver operating characteristic. This is a 3-way classification task, TA, farm, or non-farm. And we're showing the performance with respect to each target class separately. For AUPR, the dashed line, which represents random performance, is set at the fraction of examples with that class label. For area under the receiver operating characteristic, that the random line is always set to 0.5. Elastic networks implemented with GLMNet and random forests implemented with Ranger perform very well to classify TA samples from non-TA and also have some modest signal to distinguish the farm and the non-farm groups. We next inspected the features used in the elastic network models distinguishing TA from non-TA. This heat map shows top features from elastic nets as well as top differentially abundant microbes. The bottom half is higher in the TA samples and includes Bifidobacterium longum right here at the top, as well as some other less abundant distinguishing microbes. The top panel of the heat map includes features that are lower in the TA samples compared to the non-TA samples. In conclusion, our study observed that breastfeeding and traditional agrarian lifestyle influence the gut microbiome at 2 months of age. Namely, the TA infant gut was primarily colonized by Bifidobacterium longum subspecies infantis. B. infantis has a broad capacity to break down human milk oligosaccharides. It is declining in industrialized communities, but still found in agrarian communities. B. infantis and other early life gut commensals may influence healthy development by protecting against pathogen colonization and by producing nutritive and immunomodulatory molecules, including B vitamins, SCFAs, and tryptophan metabolites. I'd like to thank all of my colleagues that contributed to this project, our funding agencies, and of course, the participating families. Please feel free to contact me or Irene by email or in person at the conference. Thank you.